welcome to everyone. My name is Ray Dorsey. I'm the director for the Center for Health and Technology. Today we're going to talk about the digital dashboard. Brain disorders. Brain disorders are the world's leading source of disability, more so than cancer, more so than heart disease, and more so than infectious diseases. Parkinson's disease, a shameless plug for our book, is the fastest growing of these disorders, yet the most effective treatment for Parkinson's disease is 50 years old. Alzheimer's disease affects 5.5 million Americans. Everyone listening in has had a friend or family member affected by Alzheimer's disease, yet we have no effective treatments for Alzheimer's, none. Enter Blackfin and Derek Bowen, our guests. Huge data sets from imaging, genomics, biology, and digital devices are emerging, but making sense of them is almost impossible. Blackfin helps researchers and pharmaceutical companies see these data in entirely new ways. Derek is Blackfin's Vice President of Development and Strategy and a good friend. He combines a background in finance and capital markets with 6 years of work in the pharmaceutical industry. Today, he brings that expertise and new ways of looking at data to Chet Talks. Derek, thanks for joining us.
Hey, you bet. Thanks for having me today, Ray.
Can you tell us about Blackfin?
Yeah, absolutely. So Blackfin is about 5 years old now. We've been around for, I guess, about 5 years. And the basis of the premise was to really key in on a lot of what Ray just mentioned, in that these are unbelievably challenging diseases, which is one of the reasons why we haven't made the breakthroughs that we've seen in other diseases. And I think that there's, as we'll kind of have a conversation about, the patient journey, the data that's captured, all of that is a component of trying to unlock these diseases. And so when our co-founders got together, they had a various wealth of experience and knowledge, but they were all kind of seeing the same thing, which is in order to tackle these diseases of the brain, we really needed to break down the silos of these different data types and tease them together to understand and capture a holistic view of the patient. And when we started that journey, I don't think, to be very honest, I don't think we necessarily had an idea of where that application would be most useful. We knew that it was extremely beneficial across the entire paradigm of patient management, drug development, drug discovery, clinical development. And so as we talk a little bit more about our history, you kind of see us touching each of those points and having individuals from our kind of co-founder core also touching each of those points as well, and trying to identify where the greatest point of reach would be. So yeah, that's Blackfin in a nutshell. And we could happy to dive in deeper into some of the other relationships and what we're really focusing in on now.
So perfect. Let's talk about one of the disease areas you work on is Parkinson's disease. Can we start there first?
Yeah, absolutely. So Parkinson's has been clearly a focus for Blackfin from early on. We had started, and we can talk a little bit more about looking into epilepsy and where that had a core from where some of our co-founders were from a clinical standpoint. But as we started to look into Parkinson's, the basis was actually that a lot of these brain disorders have a lot of potential overlap from a mechanistic standpoint, and actually a lot of that is replicated for how kind of the clinical phenotype of them plays out over time. And so we started with some early work that our data science team really drove, which was around the linkage of some high quality gold standard data sets that many people in this audience are used to, identifying in PPMI for Parkinson's and thinking about ADNI and Alzheimer's, and starting to tease out if we overlap these data sets together. And there's a kind of a hypothesis there to ask, or many of them really, from a data science or clinical standpoint, to ask questions over top of those two data sets. But then there's also a notable undertaking from an engineering standpoint if I want to not only talk about these data sets and ask a singular question, which is how we've historically asked a lot of these questions, but actually modeling that data, bringing it forward and putting it in an environment that then I can go back to that data in an effective way and ask that question over and over again, or a different question on top of it again and again. And as we started to publish some of our findings and put up a couple posters at events like SFN and the like, we started to get a little bit more attention from folks who were paying attention to similar efforts, and even the folks who were sponsoring these efforts, and that led to a partnership with the Michael J. Fox Foundation, which has been a, you know, kind of a big cornerstone to where we have gotten to over the last 2 years. You know, when you partner with a group like the Fox Foundation, and then you start to build out thereafter to people like yourselves and all of the centers that are extremely well versed in these disease areas, then you add another layer to what is necessary to target and solve these diseases. So, you know, we talked about, you know, the silo that exists within these data from just the data modality that comes in, right? If I'm at the University of Rochester, my imaging data usually goes here, my clinical data goes here, my genomics data goes here, and rarely are there a lot of challenges of linking that data together. And then you try to factor in all of the other sites and efforts that are ongoing at some of these large foundations that, you know, that presents its own challenges too. So and on top of all of that, there's this kind of clash that exists with what I'll call traditional, maybe I'll just call it biology and kind of clinical mechanism, and then kind of engineering and data science, and I refer to it as the clash of APIs. So one of those groups speaks of APIs as active pharmaceutical ingredient, and it's always entertaining when we have conversations with pharma clients or biotech clients, and I'm so guilty of when I first started at Blackfin, and everybody's talking about APIs. And it's like, okay, here we go talking about ingredient versus, right, program interface, and which is all of our data scientists and all of our engineers are talking about, and getting a team that we've built internally, but then also a ton of other experts that exist within places like the Fox Foundation to give us a great deal of their time and knowledge to link together what the questions are that they want to ask, what we want to then be able to do from a data science standpoint to be able to answer those questions, and then from an engineering standpoint to be able to build the pipeline that's necessary to be able to ingest that data, link it together, and then be able to do that in repetition over and over again. And so we've built on the relationship with the Fox Foundation into a handful of other relationships that are out there as well.
Let's on Chet Talks, we like to show and not just tell. So I have some data you want to show us. And while you pull that up, I'll just give a little background. Derek's used to talking in Parkinson's disease lingo already. So he's doing a lot of work with the Michael J. Fox Foundation, which is launched about a decade ago, the largest observational study looking for biological markers or clinical markers or imaging markers of progression. That study has over 1,200 participants in 40 different countries. And Derek's going to take us through a little bit of that data and new ways of looking at data from clinical research studies that might make a little bit more intuitive sense than traditional paper documents.
Yeah, absolutely. So thank you for the intro on that, or the background, I guess, on PPMI, especially Ray. Ray certainly knows it very well, and you know, this is telling because PPMI, I think, is certainly a gold standard in Parkinson's, but it's also not the only data set that exists where people have captured information at scale, in depth, of high quality, and that goes to part of our strategy as well, which I'll touch on very briefly. You know, if we think about these data sets, there's a lot of information here, and why we focused on these data sets in the disease areas that we wanted to is actually because if we think about the data and the patient journey, which I touched on briefly, if I'm a Parkinson's patient, that patient journey is extremely different, and the data that is captured on me is extremely different than being a cancer patient. And a great example of how big data has revolutionized healthcare and drug development is clearly with what the guys at Flatiron have done. And their story is amazing, and I love what they've done. And they'll tell you that it was an extremely hard 5 to 6 years to move that forward, and that the turning point for them was really establishing this EMR and getting that into a lot of their collaboration partners and academic centers to then be able to capture that information. The fantastic thing about an EMR for an oncology patient is that EMR is point of reference for everything. If I'm an oncology patient, I come in, I think I have cancer, then I receive I receive an initial screen. I maybe then go get a genetic test. I have some imaging work that's done. I get a diagnosis, and if I test positive, then I'm coming back to the hospital probably on a 2-week cadence. And in that 2-week cadence, I'm also probably receiving some form of IV treatment that is also then all registered within that EMR. If I'm a Parkinson's patient, a lot of the information and all of those measures that are captured in there actually have a component of linkage back to how clinicians and regulatory agencies make decisions around not only what medication to give individuals, but ultimately if that medication is going to be approved. If I'm a Parkinson's patient, that journey is very different. Maybe I'm touching my provider on a biannual basis. The information and the tests that I am being scored on or scaled on tend to not always fall into the EMR, and those ones that don't fall into the EMR are actually the ones that we're approving or not approving drugs on, and there's a lot of variability or clinician benefit too, and I think that a lot of that is going to why when we started this journey, we really had a focus on these data sets, and so it clearly starts with PPMI, and with PPMI, as you can see, there are a lot of records that exist within here. And when you come into the Blackfin platform, the way that the team models data is that right off the bat, we create almost just a data dictionary of all of the data that exists here. And through this, you can really start to understand and see not only the data as it exists, but also then the relationships between the data. And that is a big driver for just our approach and everything. We, as this different data comes in, it is never Blackfin's desire to out analyze 7 bridges when it comes to genomics data. We think that they do a great job and that there are plenty of other sources that are out there, but what we want to do is we want to take the findings from that analysis and then link it into the associated imaging that might be coming. If I'm a patient at Stanford and I get some imaging work done by Kathleen, then I want to pull that down and I want to link it to my patient. And so then all of a sudden I can start to build a model around these people and start to see the linkage. Sorry.
So you got 500,000 medical records there, 70,000 biospecimens, 30,000 visits, 25,000 physical exams. I know you've done a lot of work on, you're visualizing here the relationships between many of these data. I know you've done a lot of work on progression and showing us we can look at different rates of progression in Parkinson's disease, and then that can be applied to other conditions. Can you show us how your maps of progression for Parkinson's?
Yeah, so then we kind of really start to dive into the ability to understand and model progression over time. And this is a tool that we built with the Fox team and for the Fox team initially, talking about, Ppm Progression Analysis. And again, this goes to data visualization is great for, and you and I have talked about this and other times, is that being a data scientist is a fantastic skill set and one that I am very envious of our entire team. But what becomes really powerful is when you can represent and open the conversation to others beyond just data scientists. And so there are plenty of clinicians and other folks that are out there that don't interact with data programmatically. And so how this data is historically stored is just CSV file after CSV file after CSV file, and those are updated on a somewhat regular cadence, and a lot of times people review that information in a snapshot at time. Again, going back to why these data sets are important, there's a longitudinal factor, especially to diseases of progression and neurodegenerative diseases. And so what the team here has created is what we consider almost a great filter or initial snapshot into this data set that historically, a lot of people have looked at PD in general as a, you know, a pretty heterogeneous disease, and all of the data sets on an individualized basis. And what we can do here is maybe start to tease out some of that, but more so what it allows you to do is, instead of having to go back and ask a stats team for an individualized analysis, what you can do is really start to edit and analyze questions on the fly. And so if I want to amend my, you know, my baseline, and or, you know, the range of time period that I want to look at, that's something that we can effectively do. If I want to add, you know, another filter to assess or break down, you know, the age of individuals that I want to look into, because, you know, I really want to focus on most likely earlier patients, then that is something that you can dive into. And then you can ultimately filter this group and export that information to a data set that you can then do greater analysis on or not. But, you know, what we also look at is that there's various ranges here, and this can be dependent upon what you as a team want to look at, or just what the data is offering up. And the team builds out these pretty extensive uh, offerings that you can kind of amend on the fly and see how that affects your variables. And then just down here real quickly, you know, based on how you break down your group, then you see which features have the greatest correlation or are predictive of progression on the fly. So this is just a kind of a on the fly linear analysis of tying the two together. The other thing that, you know, many people talk about, which is something that maybe it was a little bit of a learning for myself, as going from kind of a true, you know, not being a specialist and certainly not being a neurologist, in thinking about, and even a trialist for that matter, is when we talk about these diseases, you know, there is PD versus you know, a handful of other diseases. There right now is not that much of a correlation between genetics and the disease. You know, that there are certainly some early signs of genetic links that tie us to maybe 10% of the population when we start talking about LRRK2 and GBA. But what we have, even with those populations, is that there's kind of what defines people who are fast progressors or slow progressors or even non-progressors, right? There are about a third of the population that enters into a trial or comes in for clinical research really does not progress at a rate and for an extended period of time, and then ultimately may fall off the cliff. But those individuals, as we identify, can really have, you know, somewhat of a detrimental impact on interventional trials in particular, because if they never show any disease progression, then they can have a notable impact. And so how do I start to identify and tease out what makes those individuals?
So sometimes the interviewer doesn't give the guest any airtime, and other times
that's me. Yeah, sorry.
Moderator any airtime.
You are the expert.
So we're going to ask a little more punchier questions. So Derek, you have right now, I think you pulled up how you can identify people who have fast rates of progression in Parkinson's disease versus slow rates. And then we have people already asking some questions, and we're going to get to those in just a second. But so if people are looking at what's the rate of progression for Parkinson's disease, because you want to enroll people in a clinical trial who generally have fast rates of progression, whether that's cancer or HIV or COVID for that matter, or Parkinson's disease, can you tell us a little bit how you go about identifying people who have fast rates of progression?
Yeah, so it's a great question. And when we start talking about, I think that, you know, fast progression is historically defined by just these, you know, what are our traditional clinical measures, and those clinical measures then start to play out on, you know, what happens to my UPDRS Part 3 score over time, and that is, and Ray, I'll let you actually provide you know, the full detail on that. But right now, that is the main, or historically has been the main measure of disease progression for Parkinson's. And what we'll tend to see with individuals that have kind of a faster decline is you'll see, it varies actually, and that becomes part of the challenge is that we run on these averages of when people become diagnosed, have this decline curve, and that decline curve really starts to show when people start to see, and you know better than I do, right? The percentage of decline that happens over time. But the tough thing is identifying those people early enough, and this new focus on prodromal patients that allows you to almost get in front of those individuals and maybe be able to monitor and stratify them accordingly. But as we look to identify our fast and slow progressors, we are able to do, and this has been an effort that our data science team has put together on not just looking at PPMI, but pulling together multiple data sets, which I think might actually go to some of the questions that we're starting to get in here, but harmonizing multiple data sets, and then pulling down traditional measures within clinical scores to tease out and create a measure of progression that we're comfortable with and can measure beyond, and also more so, I think the more so what we're really trying to do is actually tease out individuals who are not declining, call it 10% or less, or even in some cases, I think it's 5% or less of that decline over a certain period of time. And that period of time, again, also varies depending on the study. And I think right now we're looking at a baseline to 2-year window across these 6 or so data sets that we originally pulled together.
So we already got a question from Igor Koval. He's looking at the data that you have in front of here. It looks like, for example, that slow progressors, or maybe fast progressors, in blue, had a lower severity score at baseline as measured by their Parkinson's disease. One, is that true? And then the pace of changes appear to be uniform.
Yes to both, right? I mean, I think that this is the validity, or there's validity in that, and again goes to, and Ray had asked if, so there is a form of this that we're not quite ready to show, that begins to integrate more than one data set, and this is what we start to see, and the value of where Blackfin has really grown in the last year, of integrating and harmonizing more than just one data set. So when we talk about what we learn in PD and other efforts is that it's really hard to draw inference from a singular data set. None of them really are of scale, and that historically, again, going back to trying to identify individuals earlier, understanding the transition of when they go from prodromal and transition beyond that is the baseline measure here has a little bit of variability that exists to it as well. So it's a challenging disease, and it also ties together the limitations of just clinical measures as well, which is something that I think we're starting to talk about and expanding into, is leveraging the platform's capabilities and the team's kind of open approach to bringing multiple data modalities to the table, is that the more information that I can bring when we start talking about sensors, direct-to-patient engagement, all of that is again going to this holistic picture of the patient, not just relying solely on clinical measures that historically we've utilized.
So we're going to get to sensors in a second. You look like you've integrated disease progression as measured by these traditional clinical rating scales with a biological marker down there. The second half talking about CSF levels of a protein, called alpha-synuclein in the cerebral spinal fluid, which is the fluid that bathes the brain and spinal cord. Can you tell us what we're seeing here?
Yeah, I mean, I think that very, very quickly, what we're seeing here is just the breakdown, and then actually delineation of progression as it relates to what we're seeing in blood levels for these individuals. And even that, I think, a lot of hypotheses that we're seeing in the development pipeline right now for PD are associated with alpha-synuclein and tying to that. Is there enough here or elsewhere to kind of tease that out? Maybe, or is it an existing subtype that we want to go after with alpha-synuclein? But what we're within this, and again, right, as we create this filter, we're only looking at 100 subjects, right? So it's really difficult to tie out or link to the biology. But here, what you're effectively able to see are those measures from those individuals and how we define them between fast and slow.
So if I look at this, it seems to me in this case, the level of this protein alpha-synuclein in the spinal fluid doesn't really tell us whether someone's going to be a fast or slow progressor.
I would wholeheartedly agree with you there.
And so if you want to maybe, I know there's some other things that you want to show us, either in epilepsy or Parkinson's disease, if you want to pull those out. What this really looks like is looks like what the electronic health record was supposed to look like, that we could readily see populations of patients that we're caring for and see various different metrics, including imaging, clinical data, biological data. But really, electronic medical records haven't lived up to that potential to date. But you're showing what this potential, how it could be applied for clinical trials and clinical research data.
Yes, I mean, I think that what we think is, or what we believe is of value here expands, and we talk to a lot of collaboration partners and talk about what like EMR plus could look like. What is EMR plus? They're and what a great mentor to myself and colleague to you, Carl Kieberts continues to say, right, is that the EMR system, and he's not the only one who said it, but the EMR system was very much built around the basis of payment, right? I think that what we're, as technology, again, continuing to draw the intersection between, the API cultures, as the intersection of healthcare and technology continue to grow and expand, there's an amazing opportunity to really start to tease out and link the capabilities and create a link of the patient and outcome that we haven't had before, and again, for it to be holistic. I think that the challenges, and going to what we really learned in epilepsy that I was fascinated by, not being an epileptologist, is when you talk about the breadth of data that comes in on these patients, it is unreal. Just on EEGs alone, the number of file formats that come in, the various approaches to analyze those EEGs are all fascinating and unbelievably deep. All of that, it still has created little, up until recently, benefit for ultimately those patients who those EEGs are captured on. An interesting fact is that we have all of this information within epilepsy, and all of these massive files and EEGs and imaging and everything else. But the clinical endpoint that we still use for epilepsy is
number of seizures.
Seizure diary as recorded by an individual who has epilepsy, usually, right? And sometimes caretaker, but a lot of times those individuals do not have the caretakers unless we're looking at pediatric populations. And so in that scenario, and if you read a handful of any great stories that there are out there around just the patient journey as an epilepsy patient, they forget on average, when looking back, about half of their seizures. And so we have spent a lot of time early on in our life cycle, and still continue to support a handful of clinical trials with partners around looking into secondary endpoints, linkage of other data that we can move to, and you see the benefit of actually sensors there, as we think about Empatica and the potential that devices like that have to measure actual seizure volume or seizure activity versus what somebody records. And I think in that example that they saw almost 100% increase in trials, or in seizure activity versus what people were reporting in trials, which is fascinating.
I'm going to get to sensors in a second. We got a question from John Boudreau. Can this approach be applied to existing data sets that may not be as harmonized or not harmonized?
Yeah. Absolutely. I almost wish that I could hand this over to our head of data science, Mark Hollenbeck, who's done an amazing job in building a team and an approach with our engineering team on tackling such efforts. And so, John, beyond well-established data sets like PPMI, there are plenty of them that are I'll say more woolly, and take a lot of effort to harmonize. And that harmonization process actually is something that I continue to learn more and more about and I'm fascinated by. But you can take many approaches to that harmonization effort, and it's one reason why I think that a core fundamental approach to how Blackfin operates is that we do not do harmonization, we do not do analysis, we do not do anything in a black box approach. These are efforts that have to be extremely open in the approach that we take. And when we sit down and talk with Ray and his team about the efforts that we take and the approaches that we take to harmonizing data sets, then that is a great opportunity for us to work together and collaborate over top of how we're doing that, the efforts and methodology that we use to do that. But we definitely are taking this approach and starting to link together data sets that are less known, less curated, I'll say. And there's two efforts that go into that. One, there's this kind of human in the loop approach that we take to everything, which is no matter what, and I think that most companies would tell you this, is that data curation and harmonization cannot be done entirely programmatically. There has to be some kind of human factor that goes into that. At least that's where we are now. And then on top of that, there is a data pipeline that we continue to build that gets smarter as we ingest data sets in additive data sets in Parkinson's, Alzheimer's, frontal temporal dementia, epilepsy, and start to link and get smarter about how those data sets come together. We do have some really interesting, where our background is in Parkinson's and epilepsy in particular. There's some really exciting work that we're hoping to announce on the back end of this year, some strategic partnerships that expand our reach in those areas, as well as expanding our reach into other disease areas where data harmonization is a core component of that. And I know that Ray's cutting me off, but the other effort of that is having the connectivity back to regulatory officials so that they actually buy into the approach that you're taking on harmonizing that data, so that then that data can actually be utilized for the reason why we're doing it.
We're very sensitive to our listeners. They got lots of questions. We need some shorter answers there.
I apologize.
Our great friend, Dr. Michael Geschwin, an outstanding neurologist at UC San Francisco, asks, how did you define fast versus slow progression? It looked like for here it was defined based on the Unified Parkinson's Rating Scale, but you could have defined it based on imaging, you could have defined it based on cholesterol levels, you could have defined it based on weight or any number of different outcome measures. Is that correct?
Yes. So you can select your criteria. We had, and I apologize for, again, not being the expert in this field. There were, I want to say, only a handful of measures that we utilized to specifically call out. And I know whenever we did the broader multi-data set approach, because then again, when you start talking about harmonization of data sets, what's available in those data sets, linking measure scales, all of that, I think that there were, call it 5-ish or, 5 to 10 measures that were utilized in identifying and matching those kind of fast versus slow progressors. And if Michael wants to, I'm more than happy to connect him with Mark on diving in further in that. We're talking to a bunch of Michael's colleagues on similar efforts.
And then he asked, how do you get the data? Do you get the data from an Access database, Excel, or other files? I assume all this is HIPAA compliant.
Yeah, so great question. And like everybody else, right? HIPAA compliance, GDPR compliance, Part 11 compliance, everything is first and foremost, right, top of our priority list in all efforts that we do. Most of the data that we actually capture, though, is and what I see now more is a lot of our collaborators, whether they are a foundation or they are an academic medical center or they are a biotech company, really strip out and de-identify a lot of the data up front. There are situations where they do not. And now as we move into these other strategic efforts that I was talking about, there's kind of a further effort that we have to be able to tie and link those individuals to then be able to put them into trials downstream. But this data comes from traditional sources that are big into large clinical research. So, a lot of these files are historically stored at Loney, which many people on the call are probably familiar with, that acts as a large data repository for, NIH funded and other similar research efforts, and so we access directly from them, and also utilize other can access data in many different formats. Most of the time, whenever we get data, it's coming in a massive CSV file is what we tend to see, though.
So CSV files. What statistical software you're running statistical analysis here in the background? Are you using STATA, R, SPSS? What's going on?
Yeah, so a lot of what the team works on is we do a lot of work in Python. R is very, very popular as well, and those are probably two of the main languages that or software that we're using in doing a lot of the analysis. But we as in to back up maybe a little bit, right? There's a current offer. The Blackfin platform really is actually available to, if you're an academic and you just want to post your data and start to link it effectively in the platform. You can then, the NIH uses our platform right now to do that. And for those who may or may not be familiar, there's a large effort called the Spark initiative at the NIH that is ultimately looking at mapping the peripheral nervous system and ultimately delivering therapeutics against those peripheral nerves. They utilize the platform to map and understand all different data modalities that exist out there, looking at how the peripheral nervous system interacts with the lungs, the heart, et cetera. And then with that comes different data, microscopy data, EEG data, imaging, other imaging data, and capturing all that information, and then you can map that data and then ultimately run your analyses locally as you want to, and so offer that up for anybody. It's free to do so, and you can sign up on the website.
You like we got lots of questions about cost. I love the word free. Can you upload RNA expression data? Yes, no.
So you can upload any data you want to the platform. RNA expression data specifically. So the way that it works, and I had in my notes to justify, is that you can upload any kind of data that you want. We have a full library and growing of different data types and modalities and file formats that we support, and it's very dependent upon what format that comes in, and so you can it would all be a conversation around, and this goes to, again, what we learned in our early days in epilepsy, right? There are endless amounts of time series file formats that you can upload. So it's maybe no matter what, you can upload that information, whether you can interact with it. And so when I talk about that, you can upload any kind of file format you want and store it locally, pull down the metadata from it. But then when you want to utilize some of our proprietary viewers in the cloud, those are the file formats that we support. So then, if you want to see an EDF file streaming in the cloud, you can do that on the platform. If you want to look at RNA expression data in one of our proprietary viewers, probably not going to be available, but you can still upload that information and access it and tie it to your other data.
First of two questions on cost. What's the cost for a poor academic investigator working in very rare diseases with data sets of several hundred patients?
So yeah, so right to start, if you want to access the platform and utilize what we had shared here, and being able to build this dictionary over top of your data, explore it, store it locally, tie it into. So we have a great relationship with AWS and establish the ability to push this information into localized buckets. All of that is there. It's free. I offer you to sign up and start to explore it. Where relationships begin to cost money is when you want to utilize a lot of the capabilities that our data science team is building out further. And so we have a couple different approaches that focus on data aggregation, and so we have an approach to aggregate all that information, and then what we're showing here is just a massive data warehouse, or what I'll call our data lake. And so this data lake is one that is for our research uses, and then when we want to get very opinionated about that data, it starts to come into our data warehouse, and that clinical data warehouse is where we then start to generate tools like this. And this is what a lot of our kind of clients, or collaborators, as we refer to them, collaborators or partners, start to utilize in this. What I will say is that we have finite resources, but at the same time, we have an internal R&D effort that's extremely interested in wanting to collaborate with groups like Ray's and many others who are leaders in their disease areas. And so if there's an interest in wanting to collaborate, please feel free to reach out, and I'm happy to have the conversation around some of the work that you're doing, the data sets that you have access to, or creating yourself, and actually, transitioning into what Blackfin's future is, utilizing some of the tools that we're creating to actually further increase the linkage between what I'll call kind of clinical development and data science.
So Derek does not like short answers, but Abby's going to put his email address in the chat function so that people can email him directly. It seemed to indicate that people could access the use of the platform themselves for free. Is that right?
Yes, yes.
And then how would they go about doing so? Do they have to email you, or is there a website?
No, just please go to blackfin.com, and in there there's a link that says for academics, sign up for free, and you're off to the races. There's a great list of, so as we click into there,
that's
right,
good.
right, so as we click into here, and here I'll just really quickly go to, right, if you come up here, for academia, sign up for free. And then what you'll also find here is, so right here, plenty of examples on different types of data that you can interact with. And with all of these, when I talk about our proprietary viewers, there's the ability to mark up and annotate information within the platform. So then you can generate your own metadata on the fly within those supported file formats. We also have a pretty open API that allows you to, if you want to, build new abilities to aggregate your file formats with the platform. And then what I will say last, so there's a handful of resources here on what we support. There, you can upload files, directly through the UI, or you can do it programmatically. All of that is there. And then there is a large library here of ways to go about accessing files, organizing your files that's there for people to reference.
I know I'm listed as an advisor, just full disclosure. I have no financial interest in Blackfin, so no conflict. We do work with Blackfin in Parkinson's disease, in case that wasn't clear. That's asked, how long does it take to generate a database like this? And what's the cost?
To generate a database, so right, a lot of times, and this is a little bit to move forward. So there's a lot of what we're seeing here is utilizing what I'll say is an existing data set or database. What we then is, I'll say, bring it to life. That process varies on what we're trying to do. If it's a singular data set, and depending on how the depth of data, I'll say, it is kind of all the variability. And sometimes it can take, we've done work with collaboration partners in the pharma space, linking together their data from clinical trials that has taken a couple, a week to a few weeks. We've done work with large multi-center trials and linking multiple center trials together that takes 3 months. And so there's just a lot of variability around what you are trying to extract from that study and the depth of that study to then create this.
Igor Koval asks, do you feel that users, practitioners, clinical trialists are more interested in having a clean, user-friendly dashboard of raw data, or having machine learning features that might look a bit like black boxes today?
I almost want to turn this on you because you are a routine neurologist. When you're not writing books, tends to see patients. But you tell me. I think that a black box approach is a challenging one in a highly regulated environment, and one that the long-term goal here in my mind is actually taking all of this data, and is behind all of this data are individuals who have disease. And what we want to do, and what I think a lot of the other people who have been a part of this series is actually further empower those individuals. Because, in the questions that we were thinking about in this lead up is, what continues to surprise you about this space? Less than 1% of Parkinson's patients and 1% of people with brain diseases in general participate in clinical trials and clinical research. And that's really, really unfortunate, right, for an area that continues to struggle. So how do we break down these barriers, and how do we start to feed information back to these individuals so that they come back for more? And so historically, you have a lot of people who or centers, academic medical centers, physicians that act as what I refer to them as data custodians, and I don't mean that in any derogatory meaning, but they're data custodians happily storing information for patients, and that's great. They utilize it for their benefit, but I want patients to start to utilize tools like this, like what they can engage with AI Cure, and start to interact and own their data more, and own their disease more, and participate more in this. So I think taking a black box approach to any of this from a regulatory standpoint, from a patient engagement standpoint, is the wrong approach, but that's my 2 cents.
On the patient engagement front, our good friend, Dr. Adam Dicker from Thomas Jefferson University asked, will this get to the point where the consumer or research participant can get insight into their personal situation?
You bet. Right. And I think that this is something that is a little bit of a challenge on, and we're doing expanded research now on what it is that patients want, right? And there's twofold to this. One, we're starting to see a regulatory shift that goes from what historically has just been clinician measures as outcome to actually being measures of function and patient reported outcomes that ultimately drives that transition to being more patient focused, I think. But if we look now at what's out there from a tool perspective, there's very little, if any, that gives, in particular, Parkinson's patients much feedback around how they're doing. And that sword cuts both ways, because ultimately these people have a degenerative disease, and they are unfortunately in an environment where the toolbox, i.e. medications, devices, et cetera, that's out there, as Ray queued up in the opening, is very thin. And so there's, how do I give them the right amount of information? What is the information that they want and they can utilize and actually act on in the current environment, and how does that change as our pipeline continues to grow? But I think that this is all exactly the direction we want to move in. And actually, if we think about what this data does, and what a lot of this data does, is actually people can start to target themselves, see how they're doing based on how they rate on some of these scales. They can then become informed on what clinical trials are they available for, whether that be observational or interventional, and start to move that path forward on upping those numbers in terms of, so there's 2 ways to up that number of people who want to participate. One, lower the barrier of entry, so I don't have to drive 6 hours to drive to University of Rochester to get to go to see my visit. I can do that remotely. And then 2 is really then starting to give more information and insights back to the patients to see where they sit on that scale. And then as a result of that, what can they do with it in terms of participating in other things?
So we like to make our guests on Chet Talk sweat a little bit. So you need to give them some tough questions. You're giving them lots of softballs, and
because they know that it's only veneer. All the people with the deep answers.
So how long until research participants can start to see their own data on your platform?
So we have some strategic efforts that are ongoing. We will launch some, our hope is with some great partners that we will be launching direct to patient engagement efforts in 2021, early in 2021. And there's some work there that we will then build on, that I think will start to incorporate a very informed opinion on this. So, not just looking at PPMI, not just looking at PPMI plus ADNI, not looking at just 1 or 2, but 10, 20 data sets, looking across, tens of thousands of patients, that then you have greater comfort around where these individuals sit in terms of their disease progression, and then also linking in all of the other factors that go to being a PD patient. As we do more and more research, you find that there's other information that they want. And as Ray will gladly tell you, that right now the most effective thing for individuals who have PD is exercise and diet. And we're happy to tell people that that's the case as well. And if there's information that we can push to them in that collective app, then I think we need to do that as well. So, trying to go from initially asking folks the questions that we want to get the answers that we want from a data science perspective, to then incorporate the additive value that they derive beyond just, hey, how am I progressing in my disease, but then how can I make myself better? Those are all things that we're going to build out and launch in 2021.
2 minutes for 2 questions. Make it even faster. Mary Beth Arzak asked, have you seen much value in physician notes? How could they be used in a holistic approach and to include in multiple data sets?
Yeah, so there, like anything, there is a lot of value in scribe notes. I think NLP remains somewhat of a challenge, so then a lot of that just becomes scrubbing that information, accessing that information through data. Like in these data sets, there's not as much of notes. You're looking at much more methodical scales. There are notes, but not fully. It's more so where I think we want to maybe replace physicians' notes is actually going back to this patient-reported outcomes piece. And there's some great research that the folks Gray Matter have done around running natural language processing around some of these PROs to kind of give an idea of what some of those outcomes look like and what we can pay attention to there. But there's value there. It's just getting access to it and having the resources to do it.
He tries to end, but he can't stop.
Can't.
Tanya Henderson asked, do you see a future where people can submit their wearables and lab data to see how they compare for their own health to maybe a population or to a true baseline?
Yes. We are in conversations now with many of people who want to do that. And it's a component that, so, we have some existing software and capabilities to leverage in capturing time series data. And as we continue to get smarter, we and people in general, bringing that information in, I think is fantastic. I was talking to some folks last week who are in another neurodegenerative disease who are getting ready to start a large scale patient direct outreach. And the fascinating thing is that a lot of these people can sign up, have disease for 5 years or so, or maybe 0, but with them, they actually bring 3 years of data through those sensors because they've worn a Fitbit or they've worn an Apple Watch for that time period, and then they bring that data to the party. And so having a platform and an approach to be able to do that, I think, is extremely beneficial. And I do absolutely think that's moving forward. And there are great companies, Evidation and others, who I think are also spending some time looking into that.
COVID has obviously disrupted clinical research, clinical trials. Ability to do rating scales has been diminished. How do you see data changing in a world affected by COVID in 1 minute or less?
Yeah, absolutely. So, patients with neurodegenerative disorders are at high risk, I think, and we know that. I think that the transition to telemedicine, telehealth, remote monitoring is huge, and we're starting to already see before this the buy-in, as I mentioned, from regulatory agencies to support some of these new measures. So I think that we will absolutely see that. What Blackfin is doing is Blackfin, similar to what I talked about with kind of this EMR Trojan horse that existed within oncology. Blackfin is creating this PRO platform that we touched on, but also an electronic data capture system that we call data capture for data science. And a lot of those systems historically have been used to be a lockdown system for clinical trials and support, making sure that they're locked tight, but are not all that friendly in terms of data curation and data analysis. And we're rolling that out and plan to roll that out in combination with some remote studies as well as traditional studies, and want to touch any PIs that are interested in utilizing such a tool for their research.
So, Derek, thank you very much for joining us today on Chet Talks. We have his website, we have his email. He'd be delighted to hear from you. We have some outstanding things happening in June. On June 16th, we have Senator Tom Daschle, former U.S. Senate Majority Leader, is joining us here on Chet Talks to talk about the future of health policy. June 9th, we have Chris Boone, who is the Vice President of Global Medical Epidemiology and Big Data Analysis at Pfizer. He's probably very busy in the setting of COVID to be thinking about global medical epidemiology. And maybe Derek will join and maybe ask him some tough questions.
Gladly.
On Tuesday, June 2nd, we have my good friend and colleague, Dr. Bruce Leff. Bruce Leff is a professor of medicine at Johns Hopkins Medicine. If you want to know about academic medical centers and the future of healthcare delivery, there's no better person to listen to than Dr. Bruce Leff. 25 years ago, he conceptualized that we could provide hospital care to individuals in their home. We think about COVID today, we're providing hospital level care to people with COVID in their home. He was only a generation ahead of the curve. So until next Tuesday at 12 noon with Dr. Bruce Leff from Johns Hopkins, I'm Ray Dorsey, the Center for Health and Technology. Thanks you and thanks Derek for joining us for today's Talk.
Thanks everybody.